Okay. Hello, class. So, last week we went over DNA, transcription, and translation, how proteins are made. And we went over in the structure of DNA. So you're just checking, making sure everything's working. Yes, it is. So today, what we're going to do is go over DNA replication, the cell cycle briefly, and what's called cell division, which is binary fission, in bacteria, prokaryotes, and mitosis in eukaryotes. So let's continue. So way back in the first unit, we talked about a couple of things about life. And one of those characteristics about life is all cells divide. Unicellular organisms divide in order to reproduce, and cellular division produces continuous supply of replacement cells in multicellular organisms. So in unicellular organisms, they reproduce or they divide to reproduce to make more cells. While multicellular organisms, they go through cell division to have a constant supply of cells. For example, for us, our skin is a great example of that. The top layer of our skin is made up of thousands and thousands of billions of cells. In order to maintain our skin, protect it anytime we rub it, we scratch it, we get a wound, our skin has to have cells that constantly divide. So they constantly go through cell division to make sure they are repairing. So cell division, all cells divide, whether it's a unicellular, a one-cell organism, or a multicellular, a multi-cell organism. Now, let's talk about briefly the cell cycle and in terms of different types of divisions and so forth. So, sexual cell life, eukaryotic cells divide by mitosis. A fertilized egg or zygote divides by mitosis. Mitosis means cell division. It's a specific type of cell division that produces identical cells. The fetus grows and develops into a mature adult consisting of countless cells. So what we're doing is we are looking at a specific point here. We're looking at the fertilization here going into the zygote, which is the zygote is now a fertilized egg. And when we go from a zygote to the embryo, and then eventually to an adult, we go through mitosis, which is cell division. In order to produce a full living organism, that zygote has to be able to divide its cells, make identical cells that can change, and produce the organism, in this case, us, a human, both into a baby and then as the baby develops into an adult. And even as an adult, our cells are still going through mitosis to help heal tissue, heal wounds, and help us sustain life. Eukaryotic cells divide by meiosis as well. Each mature individual produces sex cells, which are going to be sperm and eggs, by another form of cell division called meiosis. Meiosis occurs only during reproduction, and that's the key thing here. Yo. Meiosis only occurs during reproduction. So mitosis happens for development and for healing in multicellular organisms. But meiosis only produces what's called sex cells or gametes, sperm and egg. These are only used for reproductive purposes. We're going to get more into this in the next chapter next week. But no, mitosis produces identical cells that are used for growth and repair, while meiosis produces gametes, sperm and egg, for reproduction. So, mitosis has many roles that I've briefly talked about this. Mitotic cells, let me change my pen. Mitotic cell division or meiosis allows an organism to grow and develop, repair tissue, and regenerate lost body parts. Some organisms reproduce asexually by mitosis. So this is one thing I do want y'all to know for the exam, so make sure you're paying attention. The functions of mitosis or cell division is growth and development, repair tissues, regenerate lost body parts, and in some organisms, not a lot, asexual reproduction. Asexual means that they do not require another individual. They reproduce by dividing their cells, making an identical copy of themselves. There are some organisms that do this, even multicellular organisms that do this. But in general, mitosis or cell division, producing identical daughter cells, is for growth and development, repair tissues to regenerate lost body parts. Keep in mind the mitosis produces identical cells. Again, mitosis produces identical cells. So the purpose of making identical cells is growth and development, repair tissues, and to regenerate lost body parts. Mitosis in multicellular organisms produces body cells. Many rounds of rapid mitosis convert one cell into trillions. So as we go from our zygote here, and we go to a fetus, an embryo, and keep going down the line to a fetus. Mitosis is rapidly converting this one cell at the beginning to multiple trillions of cells in the end. It's going through multiple rounds of mitosis, making identical cells to produce a full living organism. So know that as of right now, mitosis is the process of cell division that produces identical daughter cells or identical cells. Its function is growth and repair. Its function is to heal and to repair lost body parts, sometimes reproduction. Now let's do the flip of that, cell death. So cell death is a part of life. Apoptosis or cell death is a process that can carve out distinctive structures during development. So yes, during development, the zygote becomes a fetus, becomes an embryo. Excuse me, zygote becomes an embryo, then a fetus. Through mitosis, the cells are dividing, excuse me, making identical cells to develop a full organism. However, on the flip of that, we have cell death, apoptosis. This is a programmed cell death, a purposeful cell death. And the purpose of it is to carve out distinct structures and development. And we'll also see more about its function later today. And we can see in the example here, look between the toes of this duck. During development, or even in different organisms, I want to erase this. I want to start over with this. Nope, this one. We're going to focus on the chicken. So, and this is the beginning as the chicken develops. You can notice that its feet are more kind of webbed. There's not individual toes. But as it develops, it's losing that webbing between its toes to make distinct toes. That process of going from more web feet to more individual, distinct toes, the cells that in between those toes go through apoptosis, excuse me, they go through programmed cell death. They are made to die and to ultimately help that structure form. Guys, this is not a unhealthy mechanism. This is a purposeful thing our cells will do in multicellular organisms. Some cells are going to go through apoptosis, programmed cell death, in order to stop development. And we're going to see that more later on too. Okay, so here's the first question for you. Shortly after fertilization, a zygote divides into two identical cells. What type of cell division produces these two cells? Pause, think about your answer. Okay, so key things here. We're going through division. If we go through division, we're not doing apoptosis. That's programmed cell death. And we're producing two identical cells. If we produce two identical cells, that's mitotic cell division or mitosis. Anyway, meiotic or meiosis produces different cells, not identical. So that means this is out. These are the sperm and egg. Each sperm that is produced and each egg that's produced is genetically different from one another. There you go. The answer is A. Now, next thing, explain the roles of mitotic cell division or mitosis and meiosis in the human life cycle. There should be an indent. Pause, think about this. Let's go. Okay, so So let's talk about this. I want to do this in a different way. Mitosis, meiosis. So remember that mitosis produces identical cells, and they're used for growth, development, and repair. Well, meiosis produces different cells, genetically different cells, or sex cells, sperm and egg. And this is for reproduction only. So those are the differences between mitosis and meiosis. Remember, this unit or this chapter, we're focused on mitosis. Next week, we'll look at meiosis. Now, before we get to mitosis, we need to talk about DNA replication. So DNA replication precedes cell division. For each of the daughter cells, each of the identical cells produced from this division, they both have identical DNA. For each of the daughter cells from this division to have identical DNA, cells must first replicate its genome, or all the cell's genetic material. So a genome means all the cell's genetic material. So all of the DNA needs to be replicated in order for both identical cells to get an equal part of the DNA, the full set of DNA. DNA replication proceeds in multiple steps. This shows you a very general overview of what happens in DNA replication. And we're going to go over each step more in depth. So the first step, strands unwind and separate. Remember a couple of things with DNA that we talked about last week. DNA is a double-stranded molecule, so it has two strands. It's in a helix, which means it's twisted, and the strands are anti-parallel. One strand goes from 5 prime to 3 prime. The other strand is going to go to 3 prime to 5 prime. So they're opposites. So the very first step in DNA replication is the strands need to unwind and separate. Next, in step 2, each strand is a template that affects and binds complementary nucleotides, A with T and G with C in the reverse. So the original DNA strands are used as templates to make the complementary DNA strand. Complementary means it's partnering DNA strand. And it does that because the nucleotides, remember A always goes with T and G always goes with C in DNA. And the last step is each double-stranded DNA molecule consists of one original strand and one new strand as a result of semi-conservative replication. So we can see here and here, we get two different DNA strands. Both have this dark blue strand, that's the original copy, and both have this lighter blue, that's the new copy. So when we say semi-conservative, that means that each strand gets one side of it that's old, one side that's new. So we're semi-conserving the DNA. And this is very general. We're going to go more in depth with this process. DNA replication builds two strands, new strands. After replication, each DNA molecule has one parental strand and one daughter strand. So that's just like how we talked about. The dark blue is a parental strand, the original strand. This light blue here is our new strand, our daughter strand. So each new DNA has one new, one old. Now, we're going to start getting into the actual steps. For each slide, I do want you to know what happens, but I want you to know the actual molecules or the actual proteins involved. that will be on the test to know the different proteins. And we can see here, this table really nice sums it up. And those are the 5 proteins that I do want you to know. But let's go through each one by one. So first off, DNA helix is unwound. And enzymes called helicase cases unwind the DNA. So that's the first molecule of protein. Helicase unwinds the DNA. The next protein, single strand binding proteins, hold the strands apart. Hold the strands apart. So in here, we already have 2 proteins. We have this helicase coming in to unwind the DNA, to separate it from its complementary strand. And then we have these single strand binding proteins that are holding the strands apart. So remember, the first type of DNA replication is just to open up the DNA. After that, we get into the next part, my highlighter. An enzyme called primase adds a short strand of RNA to each template. So in this now, we're looking at this molecule right here. And it's the same thing right here. So to start off, we have what's called a primase. Primase makes this the little green section you see on the DNA, but that's really RNA. So it builds a short little template on the DNA. That all that primase does, it wants to start the replication process. After that, we get our big enzyme. This is the most important enzyme in this process. DNA polymerase. Remember, a polymer is a big molecule made up of smaller pieces. So when we talk about polymerase, it's an enzyme that makes a polymer, specifically DNA. So with this, DNA polymerase is an enzyme that makes the new DNA strands, starting with the RNA polymers. It has, and that's the key thing too. DNA polymerase has to start with the RNA primers. And then we can see it in these 2 proteins here. Or building off that RNA primer, we have new DNA being formed by the DNA polymerase. The DNA polymerase is forming new DNA. Nucleotides are added to the 3' end. RNA primers provide 3' ends, where DNA polymerase can add nucleotides. This is where I want to make sure you understand the purpose of being, or the no being anti-parallel. Where DNA is anti-parallel. So on the bottom part, we go from 3' to 5'. That means on the top of its side, we'll go from 5' to 3'. So this, our DNA polymerase, can go from left to right, and it's nice and easy. It's following the DNA being unwound. But what I want you to notice is the other one. Let me get my pen back out. On the other side, the parent strand goes from 5' to 3'. But the bottom, the new strand goes from 3' to 5'. So that means this section here of the RNA primer is the 3' end. So we're forming DNA this way. So on this part, we are going with the helicase. As it unwinds the DNA, we are going with it. But in the bottom part, we are not. So one strand is called the leading strand because it's going with helicase. The other is called the lagging strand because it's going in the opposite direction of helicase. So lagging strand synthesis is discontinuous. The lagging strand is this strand right here. I'm gonna do that in a different color. So erase this. Again, the lagging strand is this strand right here. On the other side, DNA polymerase adds nucleotides to in the opposite direction of the helicase moving. This is the lagging strand. So remember, helicase is moving this way. It's unwinding DNA. But in the lagging strand, DNA is produced this way. Because of that, it's the lagging strand. Because we have to make section after section. It's going in the opposite direction as DNA unwinds, so we make section after section. Versus the leading strand, which is on top, and when you erase all this so we can see it. Nope. My thing wants to work. Nope. Okay. Let me get my eraser again. There we go. So the lagging strand goes in the opposite direction of helicase, while the leading strand, which is this top one, goes in the same direction as the helicase. Leading strand makes DNA nice and easy. It's going to build the DNA as it's unraveled. Lagging strand is harder because it has to make a chunk of DNA, then go back, restart, make a chunk of DNA, and so forth because it's going in the opposite direction. DNA ligase fill in the gaps. An enzyme replaces RNA primers with DNA, and ligase forms covalent bonds between the DNA segments. So the ligase is this protein right here. And what it's doing is filling in these little gaps where the primer was with DNA. So I think ligase is like the glue. It's tying the pieces of DNA together, making them one full strand of DNA. And the last thing to note with DNA, DNA replication has many points of origin. It doesn't start at one spot on the DNA. It can start at multiple places. DNA replication begins simultaneously at multiple spots along the chromosome. These spots are called origins of replication. So we can see here, these three spots are the origins of replication. So we get helicase opening up here and here. And then the other points, we get helicase opening up the DNA in other spots too. Well, after a while, they do separate the whole strand of DNA, but instead of one helicase in the middle or one at the end, we have multiple coming in to simultaneously build DNA. If there's only one helicase unwinding the whole DNA strand of one DNA polymerase, this process would take a very, very, very long time. So DNA replication has many points of origin. Now, I wanted to read this question and I wanted you to really think about it. The image at the right shows a cell's DNA before the replication. Which of the following best represents the newly created red DNA strands after replication is completed? Pause and think about this. Okay, well, let's talk about it. So, key thing here. Remember, our helicase is going to come in, unwind this DNA. As it unwinds it, we get two black strands of DNA. And then on each strand of DNA, we have DNA polymerase, primase, and all the ligase coming in to build the DNA. But they build it on top of the old strands. With that, we get a, remember, this is called semi-conservative. Semi-conservative. We're semi-conserving the original DNA. We're splitting it in half. Each new strand of DNA gets one old strand of DNA. So the answer is B. So again, the old strand is black, the new strand is red. We get one new strand and one old strand and the new DNA strands. Why must DNA replication occur before? Why must DNA replicate before each cell division begins? Pause and think about this. Okay, so remember, in cell division, especially mitotic cell division, we are producing identical cells. We're producing two cells. So if we're producing two cells that need to be identical, they both need to have the same DNA. In order for them to have the same DNA, you need double the DNA. So one cell can go to one cell, the other needs to the other. So DNA replication needs to happen for each division so that each daughter cell gets a full set of DNA. Now, let's talk about cell division, and we're going to talk about prokaryotes first. In prokaryotes, reproduction occurs by binary fission. An asexual process that replicates DNA and distributes it to two daughter cells. So asexual means it's not producing gametes, not producing egg and sperm. It's only dividing. So binary fission is very simple. What's happening here? So in the first step here, bacteria is just living its life. The prokaryote is living its life. But as soon as it goes like, hey, I'm gonna divide my DNA. So we get these two circular DNA strands. The two circular DNA strands are then pulled on either side of the cell, and they keep pulling and pulling and pulling away until they start getting this ferrule here, this division. And after a while, you get two different prokaryotic cells, or two identical eukaryotic cells. Excuse me. So all binary fission is you take a prokaryote, you double its DNA, and then you divide it out. Divide the cell in two. Eukaryotes divide by mitosis. Eukaryotic cells do not divide by binary fission. Instead, they divide by mitosis, which is similar but more complex. So mitosis is similar to binary fission, but it's a whole lot more complex. And that's because there's a whole lot more DNA involved in eukaryotic cells that go through mitosis. So replicated chromosomes condense before eukaryotic cells division. So instead of having your DNA kind of everywhere in the nucleus, remember the nucleus holds the DNA. You want your cells, your DNA to be nice and condensed. DNA in the cell on the left is loose. DNA in the cell on the right is condensed into visible chromosomes. So with that said, so you can see here, we're using yarn as the example. In the first cell right here, DNA is very loose. You can see it's spread out in the nucleus. It's like a ball of yarn. It's not really organized. It's just a pile. Versus in the cells that dividing here, we get this nice and condensed yarn, nice and condensed DNA. It needs to be condensed because it makes it easier for the cells to divide. So the first part of cell division is you need to condense your DNA into chromosomes. Chromosomes is what you may see as an X. You may see in cell division X's. The X is a chromosome. Again, the X is a chromosome. It's condensed DNA. The cell cycle is a repeated series of events. And this graph shows a cell cycle. Many different events occur in one round of cell division. And I think on the next slide, we're going to go through. So this image shows a cell cycle. And we see cell division happens only along this part. While the rest of the cell cycle, the rest of the cell's life, it's going through what's called interphase. And we'll talk about that next. So during interphase, the cell is not dividing. The cell replicates its DNA and carries out many functions unrelated to cell division. So the way I like to think about this, interphase is one of the cells living its best life. Living its best life, it's doing its thing, it's doing its function. If it's in a multicellular organism, just being there. But then when it wants to start dividing, it's gonna start replicating its DNA. And replication happens in this phase, in what's called the S phase. S stands for synthesis. So we're synthesizing DNA, we're replicating DNA to prep for cell division. Because remember, you need to replicate your DNA, have two sets of DNA before you actually divide. Mitosis is cell division. Cells leaving G2, which is this phase right here, enter mitosis, the division of the nucleus, followed by cytokinesis, which is division of the cell itself. And that's the key thing I want you to know you. Mitosis is division of the nucleus, while cytokinesis is division of the cell itself. Mitosis, you divide the nucleus. Cytokinesis, you divide the cell. Again, mitosis, you divide the nucleus. Cytokinesis, you divide the cell. We'll talk about mitosis first. Mitosis is divided into 5 phases, summarized here and shown in more details in the following steps. And for mitosis, I do want you to know the 5 steps, including plus cytokinesis, and I want you to know what's happening in each step. In this slideshow, it's gonna go nice and detail with them. So the very first step. So this is step one. I'm write it down on the side. Step number one. It's gonna be prophase. Prophase, and then when I want what the exact things I want you to know, it's gonna be this information right here. So in prophase, the chromosomes condense and become visible. That's when you get these X's right here. Spindles from the centrosome move the pole to to opposite poles. So as the centrosomes form, we get these nice strands, and these strands are spindle fibers. So the centrosomes, which are made out of microtubules, it's part of the cytoskeleton we talked about way back in chapter 1. So these centrosomes are moving to the opposite sides. This is what I call the prep phase, where we're condensing our chromosomes and we're prepping the cell. And the next step, so this is going to be step 2, we have prometaphase. In this step, the nuclear envelope breaks down. Remember, the nuclear envelope is the outside of the nucleus. Spindle fibers attach to kinetochromes on chromosomes. For this, I'm going to make it nice and easy. Can I get rid of this? Yes, I can. Get rid of kinetochores. I don't need to remember that part. So, and prometaphase, the nuclear envelope starts breaking down. And when it breaks down, those spindle fibers, those wiry cords from the centrosome are coming in and binding to the chromosome on either side. They're binding to that chromosome on either side. Metaphase. This is step 3. To prometaphase and metaphase. Number 3. Oops. And prometaphase, chromosomes are lining up along the equator, and that's the only thing that's happening. When I see metaphase, let me highlight metaphase too. I think, oh, that's not, I want green. M equals middle. So metaphase, M equals middle. So all the chromosomes are lining across the midline of the cell. All the chromosomes are lining along the middle of the cell. And that is so step 4 can happen. Step 4 is anaphase. In anaphase, the centromeres split as sister chromosomes separate and move to opposite poles of the cell. So what's happening here, and I'm going to zoom in on a picture. We look here. Remember, we had X's, and we have the spindle fibers on either side. What they're doing is they're pulling those X's apart, where you get these little parts of the X's being pulled each way. So the centromeres are splitting the sister chromatids separate. So the centromeres split as sister chromatids separate and move to opposite poles. So in anaphase, we are breaking the chromosome into half, and each half goes to a different side, different pole of the cell. Again, we are splitting the chromosomes in half, and each half goes to different sides. Now we're in telophase. This is step 5. So in step 5 is telophase. Telophase is the end. The nuclear envelope and the nucleolus form at each pole. Chromosomes decondense, spindles disappear. So this is the reverse of the first 2 steps. Reverse, of step one and two, where we are forming a nuclear envelope again. We are getting rid of the spindles. We are letting the DNA decondense. So now we have two nuclei. At the end of telophase, we have two nuclei. We are done with mitosis, but we have one more part. We have cytokinesis. Cytokinesis, again, is the division of the actual cell. In animal cells, cleavage first appears in telophase as an indentation of the cell membrane. It deepens as the ring of proteins beneath the cell membrane contracts. So when we talk about the referral, we're talking about this area right here, where the membrane starts getting pinched inwards, and it keeps getting pinched inwards and inwards until we get to new cells. In plant cells, what's going to happen instead is the cell plate is composed of vesicles. So we start getting vesicles that form along the midline here and ultimately produce a new cell wall. So in plant cells won't go through cytokinesis. They're going to just form a new cell wall between them. But in animal cells, they are going to go through cytokinesis. They're going to have that cleavage furrow and start pinching off the cell and to make two new ones. Okay, before we get to the question, I do want to go over the steps one more time. So, step one, prophase. This is the prep stage. Chromosomes condense and become visible. That's when you start seeing the X's in the cell. Spindles form from centrosomes and move to opposite poles. So the centrosomes are the rod-shaped structures right here, and we start getting those wiry lines that go between them. So we are prepping, we're getting condensing the DNA, making spindles. The next prep stage, prometaphase. But here, the nuclear envelope breaks up and the spindles attach to the chromosomes. We break down the nuclear envelope, the outside of the nucleus, so that our spindles can attach to the chromosomes. We're not moving the chromosomes just yet, we're just attaching spindles to them. We're tying them down. Metaphase, remember, M for middle. Chromosomes line up along the equator of the cell or the middle of the cell. So that's when you get all your X's, all your chromosomes to line up in the middle for anaphase to happen. Anaphase, centromeres split as sister chromosomes separate and move to opposite poles. So we are physically breaking up the chromosomes into two parts, and those two parts are going to opposite side of the cells. Now we go to telophase, which is the reverse of step one and two. Nuclear envelope and the nucleolus form. So we're reforming the nucleus. DNA or chromosomes decondense. We're unwinding the DNA. Spindles disappear. We do not need the spindles anymore. At this point, we have a nucleus. And lastly, cytokinesis. In animal cells, we get the cleavage furrow, and it just pinches off the cell continuously until we get two new cells. In plant cells, we get a new cell wall being produced by vesicles. With that said, let's do this question. In some fungi and slime molds, mitosis may occur without cytokinesis. What would you expect to find in these species? Think about it and come back. Okay. Remember, mitosis produces two nuclei, while cytokinesis divides the cell. So if we go through mitosis, we get an additional nucleus. If we don't go through cytokinesis, we don't divide the actual cell. So we don't get multiple cells, but we get multiple nuclei. That's the thing, because we are going through mitosis. We are making a new nuclei. So it's not don't contain a nucleus. The DNA does condense because it does go through mitosis. And it does interphase because we do need to replicate the DNA first. So what is the role of the spindles in mitosis? What would happen if they could not form? Pause and think about this. Okay, so spindles, remember the spindle is what attaches to the chromosome, the X. It attaches to the chromosome and pulls them apart. So that's what's pulling that chromosome apart, separating the DNA. If they couldn't form, that means we couldn't split the chromosome apart. That means we couldn't divide out the DNA anymore. And we would not divide. The cell would not go through mitosis anymore because it couldn't pull those chromosomes apart. So, with that said, the cell cycle is under tight control. The cell wants to make sure it has these checkpoints to make sure cells are going through the right steps to go through division. There's a series of chemical checkpoints that regulate the cell cycle. Checkpoints ensure that all the DNA is replicated, DNA is not damaged, and chromosomes are lined up and separate properly. So these are just some of the checkpoints. There's a lot of checkpoints in cell division and cell growth. But keep in mind, these checkpoints are there to make sure that cell is healthy, make sure DNA is not damaged, and to make sure that cell is ready for cell division. If a cell doesn't get through a checkpoint, if it fails a checkpoint, it will not go through cell division. It will halt and it will stop there. And then sometimes that cell may go through apoptosis. Remember apoptosis from the beginning of the slides. Apoptosis is programmed cell death. So if the cell has damaged DNA, it can't go through mitosis like it should. It may go through apoptosis to die. And that's a healthy thing. We don't want cells that are damaged to go through mitosis. If they do, that's what can cause cancer. Cancer cells are abnormal. Compared to regular body cells, cancer cells have distinct features. They lose their specialization, they are immortal, a- and that they can continuously or continue to divide endlessly, and they can regenerate their ends of the chromosomes. The big thing I want you to know here is the second bullet point. They can divide endlessly, and that's why we need those checkpoints. If the cell doesn't have a checkpoint, or that checkpoint malfunctions, you can get a cancer cell. A cell that just skips that checkpoint should divide, but it just keeps dividing over and over and over again, endlessly. It's a cell that's not regulated. Its cell division is not regulated. So the cell will keep dividing over and over and over without being checked. And that can cause cancer. That is cancer. And then to go back to apoptosis, apoptosis, like I said earlier, is programmed cell death. And I like the wording of that program cell death. It is purposeful cell death, so it's not accidental. Apoptosis is a set of coordinated events that dismantle a cell from the inside. And I don't want to know to know the whole process of this, but I just wanted to know in general. Apoptosis is programmed cell death. It is when a cell is programmed to die, either it's damaged, the DNA is damaged, something's wrong with it, or it's just during development, like we saw with chicken's feet. Chicken feet are normally webbed during development, so apoptosis comes in to separate the toes. So apoptosis is a naturally occurring event of programmed cell death, whether that be for the growth and development or getting rid of damaged or aging cells. That concludes today's, and then this goes over the same thing. Apoptosis eliminates excess cells, carving out structures, and weeds out aging or defective cells. That ends today's lecture. Big things I want you to know. DNA replication, its purpose, the steps, and each molecule or protein involved. You will be tested on the molecules and proteins involved in DNA replication. And cell division, mitosis. Know the steps of mitosis. Know what happens in each step. Yeah, if you have any questions at all, don't hesitate to reach out to me. Let me know, and I can help you out.